I'm going to be recording this session and we are going to make this available on the Bee City Brandon YouTube channel afterwards, as well as through the other Bee City Brandon social media. So the links will be available. We were planning on putting it up for Pollinator Week. We might put it up earlier. There's been some high interest in this talk, which is fantastic. So I want to thank everyone for coming, and I want to thank Emily for agreeing to do this talk. Emily's a master's student in the Department of Entomology at the U of M under Dr. Jason Gibbs. She's been working on a project with AFC, with myself, involving wild bee identification for the past 2 years. So her first encounter with bees was approximately 4 years ago during her undergraduate degree, which is what led her to pursue a graduate degree in the field of wild bee ecology. Yeah, it was kind of interesting. Emily started out as a student for May Elsinger, looking at forage and whatnot, and we lured her over. She got interested in bees and has taken off ever since. Now she's the person that I send photos to say, hey, Emily, I think I know what this is, but can you confirm? So we're very proud. So Emily is the current president of her department's graduate student group, has served as a youth encouragement and public outreach coordinator for the Entomological Society of Manitoba, and is also the student representative for the Biological Survey of Canada. So with that, I'm going to turn it over to Emily.
Great, thanks, Melanie. It's a great introduction. I'm just going to get my screen all ready to go here. Okay. I'm really sorry if you can hear that scratching in the background. That's my cat. He really wants in. He's very loud though, so I'm going to keep him out. All right. So I've also been looking forward to this talk for a while because I just very much enjoy talking about bees. And I will get any chance I can to talk about them. So I've developed this presentation to be an introduction to basic bee ID. And it's going to be based on my own personal experience and what I've found works best. So I'll talk a bit about things that you should look for when you're identifying bees, things to know, so some tips, especially from my supervisor, Dr. Jason Gibbs, who is a bee taxonomist, on like how to sort, best ways to sort. And then I'll touch a little bit about how to ship your specimens, if that's something that you're planning on doing. Lots of people kind of neglect this step, so it's kind of why I'm going to talk a little bit about it, just very briefly. So let's get right into this. Bee identification is a process. There's multiple steps involved when you're identifying bees, especially when you have a very large collection. So usually at the very beginning, a taxonomist is secured. So a taxonomist is somebody who describes and names species. And so they are the people who you send your bees to, for example, to be identified down a species level. Usually it's secured right at the beginning because taxonomists are very busy. There's very few of them. So you need to get one while you can. The next step is to collect. So you can do this in a variety of ways, usually pan traps or blue vein traps or nets or something of the like. Once you have your bees, then the next step is to pin, and then finally you label them. After that, you're going to be morpho-sorting your bees. So this means sorting them based on what they look like, so shared characteristics. After that, the bees are typically shipped to a taxonomist directly in the mail, so you just put them in a box and ship them off, and then they are returned that same way. So what I'll be talking about today is how to morpho-sort properly and then how to ship them. So this presentation is made for a variety of different levels, but I am assuming that you have collected bees, so you have a collection going, that you've pinned them, and that you've labeled them. So here's an example of a label. They typically include information about when the bee was collected, where it was collected. Usually they have some GPS coordinates attached to that, who collected them, and then how they were collected. And you can see that this label has a barcode on it with a unique number identifier. So each bee would get its own unique number. The barcode just makes it easy to scan and retrieve information from your computer. So you'd have all of this information duplicated in, say, an Excel spreadsheet or something like that. This information is actually very important to have on every bee that you send because it can be used to identify the bee down a species. So the flight period is useful when identifying a species. What flowering plant it potentially visited if you netted the bee can be useful. So having this information is actually a very critical step in the identification process. So my suggestion is that you do not sort your specimens until you have recorded all of this data for every single bee that you have, given each one a unique identifier and possibly a barcode if that's what you want to do, and then you've labeled every specimen. So if you have not done any of these 3 steps, then I suggest you check out the very handy manual, which is by Sam Droege. I think that's how you say it. You can just find this freely available online. It goes over everything from before you collect to collecting to pinning and labeling. They also on the website have a label making tool. So if you want ideas on how to make them, or if you want to try it out and make your own labels, I would suggest searching the very handy manual. They also have a YouTube video on how to label bees, which I suggest you check out if you haven't labeled your bees yet, or if you're planning on labeling them. All right, so I'm also assuming that you do have a box of bees that you're ready to sort, that you do not have any previous experience with identifying bees, and that you do not have a microscope. So typically when bees are identified to the species level, and sometimes the genus level, you actually require a microscope to do it properly. Since everybody's stuck at home right now, I'm assuming that you do not have access to one, but I am going to throw in a couple of microscopic features that you can look for in case you are planning on using a microscope. So handy tools that you might want to have readily available are a bright light source, so something like a desk lamp, a magnifying glass, or something of the like a jeweler's loupe. And these 2 especially are for the more tricky groups, where you need to, especially for the small bees, where you need to see them more up close to really get a good idea on them. So this will basically be a crash course in the basics of bee ID. It's not intended to replace published verified keys, but it outlines the vital steps in the bee identification process. And I do want to say that most of the photos you'll see were produced by Sam Droege and are freely available online to use and modify. They have a black background, which explains why my presentation is black. So this presentation is going to focus on the province of Manitoba, but it will work for many other provinces as well, especially across the prairies, because I'm just going to take things down to a genus level. So in Manitoba, we have roughly 360 species of bee. This is increasing every single year as more people go out and collect. These species are divided into 37 genera. This number is also going up slightly every year. And these genera are divided into 6 different families. So we'll take a better look at the genera and the families during this presentation. The problem with identifying bees is that they are very small and they can look very similar. So this webinar is going to address some of these problems related to the identification process. For example, the bees in the top row here are all different species, they're from all different genera, and they're actually representing 3 different families. Same with the bees on the bottom. So the question is, how do we accurately and quickly tell these apart? So sorting is kind of like a puzzle where you begin with this kind of jumble of species. Your bees are probably in your box based on the date that they were collected and not necessarily by the genus that they belong to. So the first step is to put similar looking bees together. So you can sort your bees by things like the size, the color, hair patterns that they have, and the sex of the bee. If you're anything like me, You do a rough sort at the beginning, but then you probably will have to go back and do a further sort. But know that it's not going to be perfect, because you're basically just wanting to roughly get things down into a genus level. It's really the responsibility of the taxonomist or whoever's identifying them down to a species level to really get that more accurate for you. So these are some tips from Jason, who is a taxonomist, like I said, on the best way to sort your bees. So his first tip is don't overfill your boxes. So instead of having your bees jam-packed into a box like this, where they're basically tip to tail, do this instead, where you leave literally some leg room. So you're not going to risk breaking any bee when you pull it out of the box. You want to leave adequate amount of space in between your specimens. It's also totally okay to have 1/3 or even 1/2 of the box empty, because what's going to happen is the taxonomist will start moving things around within the box, and so they like to have extra room to do that. You also want to have some good header labels. So what I mean by header label is basically a piece of paper that you pin into the box with your specimens that has some information about what comes after it. So for example, this header label says the genus Bombus, which are bumblebees, and I have to the right of my header label, my specimens. And you basically just want to put them in the box like you're reading a book. So from left to right, all the way down, when they're the same genus. If you have extra room and you want to start a new genus in a box, then you do a new header label. For example, here I have Nomada, and so all of my Nomada bees would be to the right of that header label. It's also a good idea to pack extra boxes, which is something that normally people don't think about, but it's very useful for the taxonomist to have an extra box to put specimens in when they're sorting. It's also okay to have a box of unknown bees. So if you have some rare genera that you're not familiar with, it's fine to just kind of dump them in a box altogether. There's nothing wrong with that. Okay, so this kind of starts our identification process. So I'm going to talk a little bit about bees versus wannabes, as we call them in the lab, which are usually wasps and flies. So some flies, for example, are extremely good bee mimics. But luckily, there are several characters that you can use to tell them apart. So what makes a bee a bee? So bees actually evolved from wasps. They evolved to visit plants for both their nectar and their pollen, which they use to feed their offspring. So that's why sometimes it can be a little tricky to tell bees apart from wasps. So bees, if I were to describe one, I would say it has 4 wings, so 2 on either side of the body. Usually they're pretty plump and hairy, although this kind of depends, as we'll see, with long antennae. Wasps also have 4 wings, and those long antennae, but they're not usually as hairy, and they're a lot skinnier. So that can be one useful character to look at. They're usually kind of bald and skinny. The exception, of course, because there's always exceptions, are the velvet ants, which tend to be very, very hairy. The velvet ants are actually species of wasp. The name is a little bit confusing. You might also confuse your bees with flies. I find that flies are the easiest to tell apart, mostly because they only have those 2 wings. Although it can be, a little tricky, sometimes wings fall off. They can be very plump and they can be hairy. So flower and hoverflies are very hairy, but usually flies are more bald, I guess. Flies also have short antennae, although there are some exceptions to this as well. So if I were to present, let's go over the face first. So wasps tend to have their antennae fairly low on their face. Some bees can also have antennae that are quite low, But all in all, wasps on average have antennae that are lower. Flies have these large eyes that you're going to look for, much larger than most bees. They also have those kind of very short nubbin-like antennae. Whereas, you know, if you're looking at a true bee, the antennae will be right in the middle of the face there, and the antennae are quite long compared to a fly. So now I have some examples. So if you have this specimen pinned and you're not sure whether or not it's a bee or something else, you can look at these different characteristics that I've just gone over. So for this specimen, I can see that it has 2 wings and short antennae. So these are fairly short here, but it's also hairy and quite plump. When you kind of think about all the characteristics together, you can come up with the identification that it's actually a fly, even though it might look like a bee. Here's another example. So this specimen has 4 wings, which you can actually see in the photo here. It's quite skinny and quite hairy. The hair is kind of a little bit covering that skinny waist, but it is quite skinny in here. And it has quite long antennae. So sometimes even I still get a little confused, and I have to look even more closely at my specimen, especially for something like this. This is actually a male velvet ant, so it's a wasp. It's not a bee. So it can be tricky, and like I said, even I stumbled up sometimes. But if you do the little checklist of, you know, how many wings does it have? Is it hairy? Is it skinny or plump? Does it have long or short antennae? Then that can help you identify whether or not you have a bee or a wasp or a fly. Now I'm going to get into some basics of bee anatomy. So we're going to stay pretty superficial here, but I'm going to outline things that you should look for and body parts that you should know. So all insects are divided into 3 basic body parts. There's the head, which has the eyes, the antennae, and the mouth, as well as like the tongue for sipping that nectar if you're a bee. The middle portion is called the thorax. It has all the muscles and includes the wings and the legs. The thorax is also called the mesosoma, which means middle body. So that's a good way to remember it. And the reason I point that out is because some descriptions of bees will talk about the mesosoma instead of saying the thorax. Likewise, the abdomen, which is this last part down here, is sometimes called the metasoma, which means like last body. The abdomen is divided into some what we call turga or tergites. So they're basically just plates that cover the abdomen. Usually there's 6 or 7 of these visible. Bees also have 6 legs. So the very front legs are called the forelegs, the middle legs are called the mid leg, and the back legs are called the hind leg. There are some features on the legs that you can look for to identify your bees. And I'll especially point out the hind tibia, which is this segment of the hind leg. The hind tibia is what holds the scopal hairs in some species of bees, so it's a good one to know. The wings, like I said, there's 2 on each side, so bees have 2 pairs of wings. The wings that are in front are called the forewings, and the wings that are behind that are called the hindwing. So the wings are exceptionally useful in determining the genus of bee that you're looking at. However, it can be a little tricky because bees are small and the characters used to identify them are even smaller. And the wings are, see-through, so they can be a little bit hard to look at. So most of these are going to be useful under a microscope. But if you get really good and you look at enough wings, you can kind of start to tell them apart just with your naked eye. So some features to look for on the wing include the basal vein back here. So I guess I should mention that the wings have what are called veins, which are these dark lines. And when the veins intersect, they create what are called cells. So an example of a vein is the basal vein, like I said. Some examples of cells would be these submarginal cells. There's usually either 2 or 3 submarginal cells, and they're numbered from the inner portion of the wing to the outer portion. So this would be the first submarginal cell, the second, and the third. They're called submarginal cells because they're literally right below the marginal cell, which is up here. And the shape of this cell can tell you a lot about what genus you have. The stigma as well, which is basically a dark spot, like an inflated vein on the wing, can tell you what you're looking at. The last character on the forewing to look for is this, what's called the second recurrent vein. I know that these names can be a little bit confusing, but if you repeat them to yourself enough, you'll be able to remember them. And then on the hind wing, we have 2 lobes that we generally look for. The jugal lobe, which is a little bit hard to see, but it's this last segment right here, and the veinal lobe. In bumblebees, for example, I think the jugal lobe is missing. So it's one way to tell it apart from something like a large carpenter bee, which would have that. This is an example of a bee from the family Halictidae, specifically the tribe Halictini. So the basal vein here, for example, is very J-shaped. If you look at other bee families, which we'll go over in a minute, the genus Andrena in the family Andrenidae has that basal vein more straight, right here. So if you compare that over here, the halictid wing, the basal vein is very, very J-shaped, like that. If you want to tell the genus Coletes apart from other genera, you would be looking at the second recurrent vein, which is very, very S-shaped, as you can see right here. It's gonna be a little bit difficult, like I said, to see with the naked eye, but if you do have a microscope handy, then this could be a great way to tell Coletes apart from Andrena, for example. So those are some characters to look for on the wing. The next thing you're gonna look at is the face. So the face, again, is divided into segments. So we have the compound eyes at the very side of the head, and these are what let the bee see the world. The bee also has 3 extra, what are called ocelli on the top of its head. 2 of them are hidden by hair in this photo. But the ocelli let the bee see light versus dark. The bee has what you can think of as an upper lip, I guess. It's called the clypeus, so it's this piece right here. And the clypeus can vary greatly depending on what genus you're looking at. It can have spots on it, so maculations. It can have hairs. It can have a whole bunch of different characteristics that you're going to want to look for. I already mentioned antennae quite a bit, but the antennae can also have characters that you can look for. So, I mean, for example, antennae are longer in males than in females. Some antennae have this first segment highly inflated, and that can kind of help you determine what genus you have. The mandibles are also something you're going to want to look at. So these are like the jaws of the bee. This, for example, is a megachile female, and megachiles are leafcutter bees, and they literally cut through leaves with these mandibles. So they have quite large mandibles with many teeth on them. So that was the basics of your bee anatomy. You can also find photos and diagrams online if you want to. Actually, I think the Very Handy Manual also has some line drawings of bees if you're wanting to know more about the anatomy. So I'm just gonna go through very quickly about some useful characters to look at when you're looking at a bee. So size, for example, is a very telling thing to look at. Most bees are quite small, so they average less than 1 centimeter in length. They can come in larger sizes though, so I kind of have these coins here as a representation of how big they are. Our biggest bees get even bigger than the size of a quarter, but these are mainly reserved for things like bombus and xylocopa, which we don't get in Manitoba, but you might get in places like Southern Ontario. So although these pictures that I'm gonna show you make the bees look like they're the same size, they actually vary by an order of magnitude. So you're just gonna wanna keep that in mind. That's one of the limitations of having a PowerPoint versus having an in-person demonstration. So usually when I'm sorting my bees, I'll start from the largest bees that I have and kind of work my way down to the smallest. You can do that too if you want. I find it works well. Something else to look for on bees are colors and markings. You can look at the body. So this is a male egg apostolon bee. And I know it's a male egg apostolon bee because it has a bright green thorax and head and a yellow and black striped abdomen. So the markings really tell me what it is. The markings can also be on the face or the legs, the antennae, the clypeus. They can really be anywhere on the bee. This is a female dianthinium. So you can, I know it's a dianthinium because it has these little yellow markings that run right along the eye there. Hair patterns, also great to look at, especially in the bumblebees. So the pattern of, yellow, black, 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 yellow, yellow can tell me what kind of bumblebee I'm looking at versus yellow, black, yellow, yellow, orange, orange, yellow. So the hair pattern can be very telling about not only like what genus you have, but also what species you have. If you are interested in doing some of your own ID for your project, I would suggest starting with the bumblebees because they are quite large. So you don't typically need a microscope to identify them. And they're cute and fuzzy. They're a good place to start. I highly recommend this book. It's called Bumblebees of North America. It has really great pictures. It has keys to males and females, and it kind of groups these bumblebees based on those hair patterns. So that's one thing to look for. All right, now I'm gonna go into a little bit about sexing a bee. So telling whether it's male versus female. The first thing you're gonna look at is the stinger and the abdomen. So people usually get very surprised when I say this, but only female bees can sting you. Male bees do not have a stinger, therefore they cannot hurt you. The stinger in this photo is right here at the very base of the abdomen. So I know that this bee here is indeed a female. If it doesn't have a stinger though, it doesn't necessarily mean that it's male. So these stingers can actually retract into the abdomen. They can also break off by accident. So stinger means female, no stinger doesn't necessarily mean male, although there's a good chance. Looking at the rest of the abdomen can also help you identify the sex of the bee. So in males, they have 7 segments that are visible on the abdomen. So those are those tergites that I was talking about earlier, these plates along the abdomen. So they have an extra segment down at the very bottom relative to a female. The female only has 6 segments. So that means that the abdomen is actually a little bit shorter in the females. This one can be a little bit tough to see without a microscope sometimes. So I suggest looking at multiple features to really get an idea for what sex of bee you have. The next thing to look at is hair. Bees are hairy and hair is very important in identifying them. So the placement of the scopy or cabiculi can really tell you what genus you have, and the presence of scopy or cabiculi will tell you that it's female as well. So it can be used in sexing the bee. In this bee, for example, it comes from the family Megachilidae, which have the scopy on the very bottom of the abdomen. So this arrow is pointing directly to the scopy, which are just those pollen collecting hairs. We can contrast this with something in the family Apidae, which have the scopy directly on their legs, so on the hind tibia, like I was talking about earlier. And these are the scopy here. They're usually very plumose and very fluffy looking, especially in the Apidae. So The presence of these two things can tell you, again, what genus it is, but also that it's female. Bumblebees are kind of this exception, as well as honeybees, although I really won't be talking about honeybees very much. Bumblebees have a corbiculae, or pollen baskets on their hind tibia. So that's basically in place of the scopy on this one. We have this pollen basket. So the bumblebee and the honeybee will collect pollen and nectar and make these like pollen bread balls that they'll stick on the outside of their legs. And you can see, if you watch bumblebee foraging on flowers, you can actually see those pollen balls on their legs. Again, just like with the stinger, the absence of scopy or corbiculae doesn't necessarily mean that it's male. And the reason is that cuckoo bees, which are brood parasites, so they lay their eggs in the nests of other bees and basically have zero parental care, lack scopy because they don't need it, because they aren't collecting pollen or nectar to feed their offspring. So this is a female Coleoxys bee in the family Megacalidae, so the same family that this bee is from. You can see that it lacks those pollen collecting hairs down here. So this is an example of a cuckoo bee. Female Hylaeus, which is from the family Callididae, lack scopy altogether, not because they're a cuckoo bee, but because they store their pollen elsewhere. They actually ingest it to bring it back to the nest. So presence of scopy or corbiculae mean that your bee is a female. Absence, again, doesn't necessarily mean it's a male. You can also look at the antennae, which are on the face. Males usually have longer antennae than females. It's greatly exaggerated in these longhorned bees, which are named for the male because they have these incredibly long antennae here. You'll notice that each antennal segment is actually elongated, which is what contributes to those antennae being so long. The males also have an extra antennal segment at the very end, just like how they have an extra abdominal segment. You have to be careful though when looking at this feature, because the antennae can actually break off. They're incredibly delicate, so if you're not careful, they can fall off pretty easily. Again, I'm going to stress that you want to look at more than one character in order to sex your bee. Finally, you'll be looking at the face. So male bees tend to have larger facial maculations, or what I call a mustache. I don't think that's the right terminology, but that's how I like to look at it. So the bees in these pictures here both have a mustache. So this very dense hair on their clypeus that kind of hangs down, looks like a mustache to me. So usually the females will have less hair just all around on their face than the males. So facial hair is one thing to look for. Maculations as well. This one is very relative because you need to know kind of what the normal is for the genus before you know how much maculations are larger, for example. So in the pictures in the middle here, this is from the same, I guess, family. This bee is a male. You can tell it's male because it has these very long antennae and it has that extra facial maculation right along that bottom edge of the clypeus there. Whereas in the female, those antennae are shorter and it lacks that extra facial maculation. Same thing with these bees on the very far right here. The male, although the antennae, you can't really tell that they're much longer, but you can tell that it has a very large facial maculation here on the clypeus, whereas the female is just this one small streak. So in the process of sexing a bee, I would recommend looking for that stinger, remembering that a female has a stinger, males don't, but the absence doesn't necessarily mean it's a male. The second thing to look for is scopy or corbiculae. Again, the absence doesn't mean necessarily that it's a male because cuckoo bees and Hylaeus also lack scopy. Oops, sorry. The third character, if you're still not sure, you can look at the antennae to see if they're longer. And then finally, the face for the mustache and those larger facial maculations. So that's a very fast description about how to sex bees. It takes practice, of course, but you get used to it the more you do it. All right, so this is the very meat of my presentation. I'll be briefly describing the North American bee families and then getting right into those genera, which is kind of the goal of your sorting, is to take everything down to a genus level. So if you are very interested in learning more about bee families, I highly recommend these two resources. So the Bees of the Prairie Ecozone has descriptions of every family. It's a good one to look at. The Key to the Bees of the Family of the Key to the Bee Families of the World by Packer and Ratty is an online, freely available description, but then also accompanying keys that I suggest you look at. So they have keys to males and females, keys to females only, but they also have a key to sexing bees. So it's a good website to check out. You just need to Google Key to the Bee Families of the World. So the first family that we're going to start with are the Apidae. I just want to preface all of this by saying I've kind of superficially grouped these bees. These are not like scientific groupings by any means. It's just more of a way to describe the variation in size and morphology that is in each family. So Apidae is the most genera-rich family, meaning that it has more genera than other families of bee. It also has the most cuckoo bee genera, which is kind of interesting. So this is the first of the two long-tongued bee families. What I mean by that is literally that the tongue is long. So the tongue can be, even as long as the body itself. If we start with this first group here, I've called it big and hairy. So these bees tend to be a larger, like medium to large size, and they have lots of hair. The most recognizable of these is bombus. So these are your bumblebees. Bombus will have hair literally covering almost every single surface on their body, whereas the other genera here usually have some bare patches, especially on their abdomen. You also know it's a bombus because, although it's a little hard to see, it has the corbiculae on that hind tibia. And I also want to note that male bees do not have that hind tibia corbicula for the bumblebees, although it can kind of look like it sometimes, but they don't. So I think I've said this a couple times before, but large carpenter bees, which are those Xylocopa, which we don't get in Manitoba, but you might get in places like Southern Ontario or BC, they look very, very similar to bombus, but they lack the corbicula. So that's one character to look for if you are in a place that has Xylocopa. The next genera that I'm going to talk about, or next genus I'm going to talk about, is Anthophora. This is one species that we get in Manitoba regularly. The Anthophora and the Melisodes and Eucera can all look pretty similar. There's one characteristic that I always look to tell them apart, and that is the face shape. So in Melisodes and Eucera, probably not going to be able to tell them apart without a microscope very well. The Melisodes has that very like ovally elongated face shape like this, whereas in Anthophora, it's much more, circular or squarish. So if you have a bunch of bees that look kind of similar like that, you can kind of look at that face shape, and that can really help you to tell them apart. Diadasia is another big and hairy apid that we get in Manitoba, but it's much more rare. And I don't know how common it is in other provinces, but I've caught maybe a handful in my university career. So if you have these bees that look similar, probably going to be Melisodes or Anthophora. Yeah, all I'll say about that. The second group of apid bees are those small and shiny Ceratina. So the Ceratina are called the small carpenter bees. They're called carpenter bees because they will actually hollow out the pith in little stems in order to make their nest. So Serotina are really interesting. They don't look anything like the other aphids. They're very small, like I said. They're like a dark blue metallic colored, and they usually have some maculations on their face as well as on their thorax sometimes. As I previously mentioned, cuckoo bees are very common in the family Apidae. These are just two examples of the cuckoo bees that I see most frequently. So the nomada right here, they're very, very wasp-like. I feel like they're the bees that people will confuse with wasps the most, but you just have to remember to look at that overall body shape. So, you know, usually they're not as skinny as a wasp. They can be just as hairless though. Once you really look at nomada and look at multiple species of nomada, you can start to understand and be able to pick them out right away as what they are. So the other group of cuckoos that we have are the Triapiolus and Apiolus. Again, probably not going to be able to tell these two apart without the use of a microscope. These ones are called your smiley face cuckoo bees because they have that smiley face on the back of their thorax. So it's actually made from a hair pattern. So these are the eyes and that's the mouth, if you guys can see that. My supervisor recently, a couple years ago, found a brand new species in Manitoba called the Apiolus gibbsy bee. So it's pretty exciting. These bees can really be, you can tell them apart from other cuckoos because these hair bands that you see are actually hair. Whereas in nomada, the maculations are coloring of the integument itself, not hair. Very interesting bees. I love those ones. I'm just gonna make sure I said everything I wanted to say. Yeah. So those are the most common aphid bees that you'll find. The next family I'm gonna jump to is the Megachilidae. Megachilidae is the other long-tongued bee family, with the Apidae being the second one. So the name Megachilidae literally means big-lipped. And this is referring to the large jaws that most members possess. For example, the leaf cutters, like I said, cut leaves with their jaws, so they need those big jaws to do the trick. The uniting features of this family are the two submarginal cells. So all members have two submarginal cells. Most other bees have three. Again, that's going to be a microscopic character. You're probably going to need a microscope for it. Females, unless you're a cuckoo bee, will also have scopy along the underside of their abdomen. I'm just trying to get there it is. Okay. Megachilids are usually described as being cigar or even submarine shaped, which I don't know if I just define it like that, but they are definitely plump looking. Common members include the leaf cutters. So these are a female and a male leaf cutter bee in the genus Megachile. The females, again, they have those abdominal scopy. They're black all over. They're usually quite large, although you can see some fairly small Megachiles as well. And they have generally some hair bands along the abdomen. The males can be interesting. So the males can either look kind of similar to the females, or they can look vastly different, like this specimen here. If you ever see a bee that has these front legs that are greatly altered with these like long fringes of hair along the front, you know right away that you have a male Megachile because there are no other bee that have that feature. And the reason for this character is that the male Megachile will actually cover the eyes of the female Megachile during copulation. Nobody really knows why. It's just what they do. The other larger black megachilid that you'll see are the Coelioxys. So these guys are cuckoo bees, meaning that they lack any scopy. So they're generally not as hairy as the megachile. The Coelioxys, I think it literally means pointy bum, something like that. The abdomen is quite like pointy instead of being more round like other bees. So usually nobody has an issue with picking out a Coelioxys because they're so interesting looking and different looking. The second group of megachilid bees you're going to want to look out for are those smaller black ones. So there's 3 different genera here that can, again, be fairly tricky to tell apart if you don't know what to look for and you don't have a microscope. So those are the Hyriades, Ophitis, and Ashmediella. Ashmediella is more of a rare genus, so you'll probably see Hyriades and Ophitis more often. It's, you know, totally fine if these guys get grouped together because, like I said, the taxonomist will be able to separate them because they have the know-how in the microscope. These ones can kind of look a little bit like tiny megachiles. It's a little hard to see in this photograph, but they do have that abdominal scopy right under there, although the leg is kind of in the way. The third megachilid group are the metallic megachilids. So this is usually reserved for osmia, so they can be dull metallic blue, bright metallic blue, they can be metallic green, so they come in a variety of colors and a variety of sizes too. So we have usually small osmia here, which are the most common, but you can get larger osmia as well. They can be quite hairy, but underneath all that hair, they are actually metallic. Although I should say there is an exception. There are some species of osmia that are totally black and have no metallic reflection whatsoever. But most of them do have some sort of metallic blue. These, I guess I should mention also, are called the mason bees because some of them will actually use mud and other material to create nests, which is pretty cool. The final group of megachilids are those ones that have large or small maculations on them. So the ones that you'll see most frequently are Dianthidium as well as Anthidium. These can be, again, a little difficult to tell apart sometimes. The Anthidium on average are a little bit bigger than the Dianthidium. Just like with the Nomada, these ones have a tendency to be grouped with wasps, just solely based on their coloration. But if you look quite closely, you can actually see that the Dianthidium and Anthidium have abdominal scopy on them. This one in the Anthidium picture is full of pollen. So that's one character to always look for when you have a bee that kind of looks like this, with those large yellow maculations on a black abdomen. They also, I don't have a picture of the face up, but they also have maculations on their face. So it's good to look for too. Another tiny, usually tiny genus of megachilid are the Stelis. So the Stelis are small and black, and they usually have some amount of maculation on their abdomen. These are a cuckoo bee, so again, you're not going to find any scopy on them at all. Again, another one that can kind of get confused sometimes with flies or small wasps. So just look for the, that hair on them, I guess. They're not that hairy, but they have quite a bit of hair on their thorax compared to wasps. All right, so that is the family Megachilidae. The third family we're going to get into are the Polyptidae. So these are, the first group is the black with the hair bands, and you'll find 2 different genera here, the Lasioglossum and the Halictus. So these ones can be very difficult to tell apart, and I have an extra slide describing how to tell them apart, so I'll leave that for then. Most halictids are small and dark, with or without hairbands or metallic color. So Lasioglossum is this huge genus. They have metallic thorax and head sometimes, as well as this non-metallic abdomen. There's one species of Halictus that can be confused with Lasioglossum, aptly called Halictus confusus. You're probably going to lump them together. I do it too. it's easy to not be able to tell them apart. Halictids can also be metallic, so we have things like ogochlorella, which are generally smaller than agapostemon. The females of agapostemon tend to be bluey green. The males, I described this earlier, but they have that green metallic color on the thorax and head, and then a black and yellow abdomen. Super awesome bees. They're spectacular. They're great to see in the field. People love them. I think they're up for Brandon Bee of the Year. If you haven't voted for that yet, go vote. The last group of halictids are those small dark ones with orange or red abdomens. So if you have a bee that is really, really tiny and has an orange abdomen, it's going to be one of two species of Lasioblossum. So those ones are very easy to tell apart. The Sphacodes are a cuckoo halictid, and they have this bright red abdomen, and the rest of their body is black. Again, they lack any kind of scopy for collecting pollen because they are cuckoo. All right, so we're gonna revisit Halictus and Lasioblossum up here. So Lasioblossum has been described as a nightmare taxon, which is fantastic. There is one character though that you can look for to easily tell them apart from the genus Halictus in the same family. And that is the placement of hairbands, if the Lasioblossum has a hairband. So in Lasioblossum, the hairbands are at the base of the turga. So in this diagram, if this is the turgite, the hair is at the very, very base. Versus Halictus, where the hair is at the very tip of the turgite. And this is true for both males and females. You can kind of see it here. So in the male here, that hair is at the tip of the turgite. Female, same thing, whereas this one is at the very base. And in the Lasioblossum, that proceeding turgite can actually cover the hairband to a degree, and that doesn't happen in Halictus at all. All right, I know this is quick. The next family is Andrenidae. So we have basically two different forms. We have black, usually pretty big, possibly with hairbands or a red abdomen. So the Andrenidae are actually the most species rich worldwide. And they come in a lot of different forms and shapes and colors. One thing, if you're looking at the face of the bee, you can tell it apart from things like Coletes by the shape of the face. So Andrenids usually have a very square face. You can also, if you have a microscope, look for facial phobia, which are basically depressions along the compound eye filled with velvety hair. Again, it can be a little bit difficult to find that. The second group are the small Andrenids that usually have maculations. So here we have three different genera in Manitoba. We have Callioxis, Perdita, as well as Protandrina, which is, I guess, formerly known as Pseudopenergus. That's not quite published yet, but that's kind of what our lab is showing. Anyways, these ones are small, usually black, and they usually have yellow maculations somewhere on their body. So it can actually be on their face, like in these pictures, or along the abdomen. Some species of Perdita are entirely yellow. So if you ever find a specimen like this, you know right away it's a Perdita. It's pretty cool. The Protandrina are fairly rare. You're probably not going to see a whole lot of them. They're very nondescript. They're just small and black, sometimes with maculations. All right, so this is the fifth of 6 families, the Coletidae. This family only has 2 genera, so Coletes and Hylaeus. The Coletes are also called the cellophane bees. They have that S-shaped second recurrent vein, like I showed you in a couple of slides ago. They also usually are quite large. They can be small. They're black and they are hairy, and they can have those hair bands on the abdomen as well. What you're going to look for if you think you have a Coletes is a heart-shaped face. So this is what stands them apart from Andrena. Coletes also has a forked tongue, although this can be kind of hard to see if you don't have a microscope and if that tongue isn't sticking out nicely for you. But they are the only genus of bee, or this is the only family to have a forked tongue. The other genus is black, it's bald, and it's maculated. It's the Hylaeus. So these are called the yellow masked bees, and they quite literally have like a yellow mask on their face. They have either a face that's mostly yellow, or if it's female, they'll have yellow beside the eyes. And so if you get these, they're pretty, although they can look like, you know, flies or wasps at first, once you get a closer look and see that face, you can be pretty certain that you have a Hylaeus. I just put this picture up because some have these like greatly inflated antennal bases here, which are pretty cool. Yeah, and again, they're bald because they transport pollen within their body instead of on their body. The last family that we have is the Melitidae. This is by far the smallest. I think there's only 3 species like in North America in total. We only get 1 genus in Manitoba, and there's only 1 known species so far. That's Macropus nuda. These again are fairly nondescript bees. They are black. They have scopy on their hind legs. Yeah, easily overlooked. Very rare. Probably not going to collect a whole lot of these. I think I have maybe a handful. They specialize on a flower called Lysmachia ciliata. And so they can only be found wherever this flower is found, which has contributed to the rarity of this bee. An even more rare bee is the cuckoo of the Macropus nuda. And it's actually been on these Costawick, it's had a Costawick status report on it. It's the Epiloides piloculus, the Macropus cuckoo bee, which is in the family Apidae, and it is considered endangered as of 2011. And that's because the populations of these guys, the Macropus nuda, are very small. So it makes sense that, you know, if this bee relies on this bee, that this bee would also not be highly abundant. So that is all 6 families of bees. That is a crash course, like I said. I'll present some resources for further reading at the end of this presentation. And so this is just an introduction. I would recommend going and reading further into the genera that I said, into the families, and even an extra genera that you might have in the province that you reside that I haven't specifically talked about today. So I have some, I think we have some time. I'm just gonna go through a couple of these really quickly. So if you have a bee like this, you may or may not remember the name. What you can look for are the presence of scopy, which I can see are under the abdomen, meaning that it's a megachilid bee, so it's from the family Megachilidae. And it has those heavy maculations on the abdomen. So if I were to go back and, you know, read about bee genera in the family Megachilidae, I would find out that it's a Dianthidium female. If I had this bee, I would look for the coloration and the facial hair. And so this is metallic blue with that mustache and the long antennae. Doesn't have any scopy, probably gonna be a male, probably gonna be an osmia, which it is, it's an osmia male. So this is just to give you an idea of what you should be looking for when you look at each bee individually. This guy, it's going to be fairly large, it's black mostly, pretty hairy, face looks like, you know, it's pretty square. I can't really see them very well, but I think it probably has facial fovea. So, and the scopy on the hind leg here. So it's probably going to be female, and it's probably going to be anandrina. Again, you might not get it perfectly sorted out, but the idea is to get a rough sort. If you look at this bee, I see in the face that it's very heart-shaped, that it has hair bands, and that it's pretty large and fully black, probably going to be a Coletes. So it's a Coletes female. This is kind of what you would do for all your specimens. This guy is, well, it's male because it has long antennae and a facial maculation. It's also very black all over, and it has those hair bands, although they're a little bit faint, but you can still kind of see them at the very base of the turga. So I know right away that this is going to be a Lasioglossom. It's a male Lasioglossom. Okay, so that's, I think, all I have for the bee genera and the bee families. Now, like I promised, I'm going to talk very briefly about how to ship your specimens to your taxonomist. So the first step when you want to ship them is to make sure that your specimens are extremely secure, meaning that the pins are pushed nicely down into the foam in the box that you're keeping them in. You don't want them coming loose and flying around all over the place. You can actually put a cotton ball in the corner, which will catch legs or heads or abdomen that tend to maybe fly off during the shipping process. It happens. You also want to put a piece of cardboard on top of your pins, and maybe even a little piece of tape, like here, like this person is doing, to easily pull off that cardboard. This is to make sure the pins stay pushed down into the foam and they don't come out. Do not use foam on top of the pins. You don't want your pin heads going into the foam and being pulled out when somebody tries to lift out the lid. So that's the first step, make sure they're secure. Second, tape the lid shut. So you don't want to risk any lids coming off during shipping. You can do this with tape, or you can do it with string or rubber bands, whatever you have on hand. Make sure that lid is secure. Next, attach a shipping address or an address label from where the bees are coming from onto the smaller box holding your specimens itself. And this is in case the larger box that you put your specimens in case that address label comes off or gets damaged or whatever, they can open it up and still have access to where the shipment came from. What you don't want is your bees to get lost in the mail forever. So make sure you include ample address labels. You can also, this is optional, but you can bind multiple boxes together so that they're more secure during shipping. The last step is to find a fairly large box where you can fit several inches of room on either side of your specimens. Fill it with peanuts and then slap a fragile label or many on the outside. And so the peanuts are to protect those specimens from getting bumped around and damaged. Also remember to fasten your address label on the large box too. All right. So these are some of my favorite resources. I have actually the first resource, The Bees in Your Backyard. It's a book. You have to buy it in order to access it, but it's lovely. It has so many different pictures. Its range is all of North America, meaning that they talk about bees from Mexico too, which isn't super helpful for Canada. But they do have keys and descriptions of all families and genera. If you want something that's freely available online, I suggest The Bee Genera of Eastern Canada by Packer et al. It's a great resource. Here's the website that you can find it at, the Canadian Journal of Arthropod Identification website. So this manuscript it's loaded with pictures of bees and descriptions of each genus. So if you want a great place to go and review what I've talked about today, I would suggest this paper here. And finally, this just came out. It's a field guide to the bees of Ohio, but it includes every genera that at least we get in Manitoba, with some additional microscopic features if you have a microscope handy. So I highly recommend this one too. All you have to do is Google Bees of Ohio, a field guide. And that is the end of my presentation. So I'm very, very happy for the turnout, and I hope that you have found this very helpful. I'm gonna stop sharing my screen now. All right. I guess we can take questions, right, Melanie?
Oh, epic. Yes. Oh, I wish that you could hear everybody applauding, because I myself found that talk to be very helpful. You were very clear on those and gave us a really good start.
Thank you.
So we do
yes.
have some questions. Yeah. So May is asking about bumblebee color, about the unpredictability about it. But you did mention that can be a place to start.
Yes, if you can get your hands on the bumblebees of North America, I think it's called, they actually have diagrams of all the variants for each species. So I know you're probably thinking of Bombus rufosinctus. they'll have every single kind of color hair pattern variation for Bombus rufosinctus in that book. So I find that really helpful because they can vary quite a bit depending on the species.
Okay, yeah, and I guess also that's where then the microscope is what comes in handy, where you're in the size of the cheek and all that kind of good stuff.
Yes, definitely. Yeah, bee identification is not easy. It's not something that really comes naturally. You do have to continue working at it and practicing it in order to get better. That's just the nature of it. Looking at pictures online can really help, you know, especially if you don't have access to a lab right now to see your bees.
For sure. Now, I know most of the people on the line are those that actually are working in this field in some capacity. Do you have any suggestions for people who want to do it just as a sort of as a hobby? I know I've been asked that before about, you know, do I net them, or what's the best way to have as low impact as possible on them?
Netting is always great for that. Whenever you use things like pan traps or blue vein traps, they can catch quite a bit of bees, more than you probably need, especially if you're just interested in looking at them up close and learning more about them. I would suggest taking, you know, a net, one that has a fairly small mesh size, out into the field with you. So what you would do with that is you would find a bee that's approaching or resting on a flower, and you would gently net it off that flower so that you don't damage the flower or the bee. Then you'd make sure that the bee is in the very tip of the net, and you can kind of, if you like hold the netting shut, you can kind of keep it there. And that's how I like to look at them more up close. Another thing you could do is take a Tupperware container out with you, a fairly small one. You can poke some holes in the lid, and then you can actually trap the bee in the Tupperware container very gently. You don't want to hurt it. Close the lid, and then that's a great way to view them up close. And once you're done looking at it, you can just release it back into the wild, and it's fine.
Great. Now, Cheyenne was asking about bees with the white butts. Can we just assume it's a gypsy bee, because we don't tend to get those kinds here, or that we don't get any other ones with the white?
Gypsy bee. You know, I'm not super familiar with the white-tailed bombus because, like you said, we don't really get very many here. But I would probably not assume that right off the bat. I would probably look in a field guide of some sort to be aware of the other kinds of white-tailed bombus that might occur in your area, and, you know, double check. There are going to be other features that can help distinguish it. So that would be my suggestion.
Okay. Does anyone else have any other burning questions for Emily? She went over a lot. I know it's a lot to take in.
Since this is recorded, you can watch it as many times as you want to.
Yes. Okay, well, if no one else has any other questions, I just want to thank Emily again for putting that together and for making it available for people to view. I mean, like I said, I think I learned a lot, and that was fantastic. Very clear.
Thank you very much. Thank you, everybody, for joining in and listening.
Thanks, folks. And as I said, we were recording it, so we're going to make it available through the Bee City Brandon YouTube channel and on the Bee City Brandon website as well in fairly short order. And we are planning on putting out a few more webinars like this, so stay tuned. I have your email now, so I'll include you on the notification. We're looking at doing some real basic fieldwork stuff, but then also looking at some exploration of native plants and pollinator supporting plants and going from there. All right, folks. Thanks again, Emily. Thank you everyone for participating.
Bye.